Thank you. Thank you. And thank the other speakers. It's really been a fascinating evening. And really, I'm going to be changing gears again. So what I'm going to be focusing on for the future is really, I've been surfing for the last, surfing the frontier of technology development since the 80s, but really the 90s. And there are two main areas that are coming together that I've been very fortunate to be a part of. And one is video technology, video game technology, and cinema technology, or derived from computers, but also what's happened, the impact of similar sorts of technologies on medicine and discovery in science. So what I do, I've been working at the Walter and Eliza Hall Institute, which is just opposite Melbourne University, behind the Royal Melbourne Hospital. This is the building here. This 6-story tall LED curtain is called the Illuminarium. We installed it 5 years ago. And what it's doing is playing a non-stop roster of raw images, raw data from the Institute. And the Institute's a center for basic research into various forms of cancer, infectious diseases like malaria, autoimmune diseases like diabetes, and it's had a massive amount of success over the decades. And what I do is, I'm a cell biologist by background, microscopist. I'm resident there. I'm trained to interpret and understand all the forms of data that they're making, but I'm also from a generation that grew up with computer games and computer graphics, and I've been doing that as a hobby and as a career for certainly since the 90s. So I started at the Walter and Eliza Hall Institute at 1995. I was their first Photoshop guy, and I evolved it into animation. So I'm going to quickly blast through and play hopefully more animations which of the molecular world, which I think you'll find mind-blowing. I'll begin with one. This is a simulation. A lot of the work that I'll be showing you is an emulation, the look and feel of a simulation. Simulations are very computationally expensive. They take a lot of very high-powered computers to do. These are water molecules, and this was created by Mike Kuiper. He works at CSIRO Data 61 right here in Docklands, and he's created a number of visualizations for me. This is water, and what you're about to see is a transition from water to ice. So all of the molecules are jiggling around very rapidly and banging into each other. And this is at 0 degrees Celsius, and through this simulation, because of their the bonds and electric charges around each of the water molecules, so the oxygen is red and the two white atoms are hydrogen. What you're seeing there is a transition from the disorder of water forming into the very ordered structure of ice. And so by looking at this simulation of water turning into ice, I can ask you, can you see why ice floats? Why is it lighter than water? And it's because of all that order, that space that happens, because of the crystal structure of water. So simulations like this very much inform my animations, but they're also, because they're so computationally expensive to do, I emulate the look and feel, but I have a lot of tricks that I can use to shortcut that. And also I have a lot of control I'm using the same animation techniques as I use in high-end video games or in cinema for Hollywood. I have access to all of those tools. This is a very small, which I'll turn the sound off. We don't need sound today. No, we don't. Okay. This is a simulation, an emulation that I've done of the molecules in your blood. So when you strip out all the water, these are what protein molecules look like. So these are all individual proteins. And what you have here, this green sphere, is actually the rhinovirus, the common cold virus. It's a very ordered geometric form. These other proteins here are called fibrinogen. And then they're a protein that lives in your blood. And when you get a wound and they get exposed to oxygen, they deploy and create a tangle, which is blood clots. So this is a preloaded blood clot. And the virus capsid, and then these are all the typical blood molecules, including antibodies. So when you hear about antibodies, they're the Y-shaped ones, which I can't see one there. So this is an emulation, and I can cheat a lot to make this sort of a product, but it's still computationally expensive to create the image for a computer because of the shadows and the calculations of collisions and so on. So it's been a very intense task. I've been involved with, I collaborate on projects all around the world. One of the really exciting ones I'm working on is with a high-end science center in Sweden who have built and it's actually launched this year, an 8K, so higher resolution than IMAX, 360 3D dome. So just like they have a dome at ScienceWorks here, which is I think 6K resolution, these are 8K. Just you won't be able to see any pixels, it'll just be like a photograph, and it's full 360 3D. And so they want to create these molecular worlds. So I'm not going to show you all my earlier stuff. I'm just going to show you what we're developing right now. But they wanted to create this sort of a spectacular show. Why? Because 100 million school kids go through domes every year, and almost always they're seeing star shows. And so rather than going out into the outer world, we're going to go into the inner world of looking inside our bodies. And that's kind of their vision. So I was very excited to be a part of that. This is in 2014. So you saw an excerpt, a close-up of this particular animation. So again, these are the blood molecules. There's your antibody there, and there's the rhinoviruses starting to appear. And this little simulation I'm showing here just runs for about 60 seconds or so. To do a 6K resolution, a high resolution computation of all the shadows and all the calculations, I had to go to a supercomputer, and it actually only took about 9 minutes on this supercomputer, but that, just for this movie here, cost about $600 of time on that supercomputer. Or I could render it on my local machine and I can wait 10 days and see the results. So I could do it quickly, or I can do it. And so for this movie of 8K resolution, yes, first of all, was it technically possible to create that many molecules and fill up the world and create the imagery? Yes, it was, but we're going to have to budget hundreds of thousands of dollars for computer time to do it. So this is in 2014. 2015, I went to a lecture like this. And what you have here, this is actually a data set created by Graham Johnson in the Scripps Institute. And what these are is, again, blood molecules. He's done them in green here. And this is actually a slice through an HIV virus particle. And these are all the various molecules you've seen it. So it's virus and HIV in blood plasma. But what you're seeing here also is this movie, looking every bit as good as mine, running in real time on a laptop because of gaming technology, because of the development of that hardware. And so I saw this and went, years of work, I just went, dump, this is where we're going, this is what we're going to do now. And so I've been working with this lab in Vienna. They're developing this technology, but also I have a guy, Justin Muir, who's from 20 years in the games industry, and he's developed a pipeline for us so we can access this kind of capability. And this is a normal gaming PC that every teenager would love to have on their desk, running like this. So spectacular stuff. So this is not only opens up possibility of doing these wonderful dome movies, but it's also for virtual reality, it's for mobile phone augmented reality, it's for all of that. And it's all in real time. So it's a really profound change of how the audience will see and experience this. It's not a finished movie, it's a world you can take your time and explore or travel around in as you wish. So that's what we've been developing. This is the technology we're sort of tapping into. This came out at the beginning of 2017, Horizon Zero Dawn. It's a blockbuster game on the PlayStation. This is all real time. This is how it looks in the game. If you haven't seen it or what PlayStation games are like these days, this is all a world. It's an open world. You can go wherever you like. And this is actually the stories in the future after the apocalypse. We've been reduced to these tribal kind of people, and the AIs have taken over, and we are hunted. And so you very much feel like you travel the world. You're trying to control and avoid all the computers and the machines, but all the landscapes sort of reverted back to a natural state. So anyway, open world, all of these details, all of these plants. This is 2016 technology because this came out in 2017. And so the question is, why can't I access this sort of technology? This represents over $100 million worth of development for this one game. It's a blockbuster game, and they made they make more money out of these things than they do if a blockbuster is successful than they certainly do out of cinema. So it's a massive industry. But they're investing huge amounts of money in the technology, and now we have access to exactly this kind of capability. The story I'm going to talk about is one of the topics we're working on right now, which is when you are breathing air and you're taking in oxygen, and when you're eating food and you digest it and you break it down, what your body's actually doing is stripping it out, and then at the very last steps, it's ripping out electrons and using them to generate chemical fuel that makes powers all over your body. And this is sort of a classic 101 biology topic that all high school kids have to learn. And it's a mess. Actually, I should have an image here. Oh, I see. We'll probably come back to a figure that give you a sense of what school kids are currently facing with trying to learn about respiration. This is actually time-lapse microscopy done at the Walter and Eliza Hall Institute where I work. You have two cells, so this is greatly sped up. You have two cells. These big spheres here are actually the nucleus. These two here, that's where the DNA is. But these little wriggly worms is what we're going to focus on. They are called the power plants of the cells, are mitochondria. They're often referred to as power plants or power generators inside the cells. And this is where the chemistry I'm about to show you is happening in these very dynamic, wormy-like, and they're branching and touching and changing all the time. And what we're going to try and do is zoom into them and show the machinery that we know about. And so why I'm showing you all of this is there's a tsunami of data coming in. We are overwhelmed with the incredible things we're discovering, but it's very, very hard to explain it to other people. But it's very easy. If you show people what it is, they get it just by watching it in action. But this is all very, very, very small stuff, smaller than the wavelength of visible light, the world we're about to go to. And this is actually also a mitochondria. This is done with an electron microscope. So it's a very, very thin slice. Like if the mitochondria was a cucumber, what you're seeing here is like a slice through the cucumber, a very, very thin one. But what you're starting to see is there's a lot of ordered structures. These tubular structures are actually membranes within the mitochondria. And the tiny little granular dots you see there are actually protein molecules. Just about see them. And you can see sort of there's ordered structure. But this sort of is just one form of data, very high resolution, compared to the colorful time-lapse I just showed you. And so I can combine and mix these sorts of forms of data and reinterpret them in animation. And this is, a classic figure from the high school textbook. So this gray bar here is actually the membrane. So I was talking about membranes before. They're if you imagine this table as a membrane, all these things are populated much like the forest was, in soil. So this is sort of like the surface of the membrane, and these proteins are embedded in them. And what all this complicated chemistry is step by step. This is how school kids learn it, and university students learn it, of the breakdown of one chemical reaction, and it cascades through all of this. And then this is actually the machine we're going to focus on just to cut to the chase. We're just going to focus on ATP synthase, this last step here for tonight. And ATP synthase is, we've known a lot of detail about it since probably the late 90s and certainly early 2000s. We have a lot of discoveries about it, this last structure here. And it's also by my peers, by far the number one topic that everyone's had a crack at. So I should be able to show you in a minute. This is from a paper that just came out about 2-3 weeks ago, a review paper covering, again, these are sort of the molecular detail of this engine. Very ordered structure. This is their diagram figure of it. And this is an animation that they generated and published it as a figure as part of the paper. And what you're starting to see is these ribbons here are representing like a backbone of atoms. And this is a rotary engine. And you can see it changes the shape. As this axle rotates, it's changing shape of the whole molecule. And these are examples. There are dozens and dozens of examples on YouTube. These are probably the best, I think, that you could find. Graham Johnson was an early pioneer. He's a dear friend and a peer who does amazing stuff. But this is a very early time, in 2002, of ATP synthase. I mean, you can do it one way, you can flip it over the other. It's a reverse direction polarity of the way he's looking at it. BioVisions is very, very famous. Probably, if you think you've seen some of my animations, it's probably by BioVisions. They're very, very high-end production studio, very expensive. They do beautiful work for Harvard. And then Alistair Stewart, who works up in Sydney, who's been advising me, he did this sort of much more accurate molecular visualization of ATP synthase. So what I'm saying is this area has been tackled by many people. These sorts of molecular details have been tried to be represented. So what new could I bring to this is kind of the idea. And this is typical of my work. I'm very, very happy. I usually don't look at what anyone else has done. It's not very informative to me. I hit the literature. I review the literature. It's a big literature review at the beginning, and just look at really what do we actually know, and that's where I build all my work from, and assemble from any form of data that we may have in the literature. This is my comparable version of what you just saw. This is an early test. This is all now in Game Engine, so it's real time. And what you're seeing is the stepping of the rotary engine. There's a lot of other molecular dynamics on the sort of jiggling nature of it that I'm able to add. And now you're starting to see a bit of the chemistry. This is where it happens. And I'll show you the final animation, and we'll get into the details of what actually is going on with ATP synthase. It's a rotary engine. So you have 10 trillion cells, human cells, inside your body. Each one of them has, thousands, it would have probably from 1 to probably up to 100 mitochondria. Each one of those mitochondria would have thousands, if not tens of thousands, of these ATP synthase in each one of them. So you have hundreds of thousands of those in each one of your 10 trillion cells. So back to the mitochondria, this is again that slice, and this is sort of the landscape of where ATP synthase, it's embedded actually at the tips here, where at the folds of where the membranes are. So from that slice, that electron micrograph slice, if you take a mitochondria, and this was done in Sweden here, what they've done is gone slice, very, very thin slices, and they're reconstructing in 3D the tubular nature of the membranes inside the mitochondria. Digression I'm not going to go into today. The blue areas are actually the genome of the mitochondria. This is DNA. These are packed little balls of DNA that, because mitochondria are actually endosymbionts way back in evolutionary history. And this is the remnant genome they still have inside them. And they divide, and they're really like living organisms inside our cells. Very reduced DNA. It's only 13 genes in there. So that was a 3D reconstruction from lots of little slices. And so this is actually an example of that, where they're stepping through slice, slice, slice. And so this engine that I was showing you before that was rotating, so that you can see it's sort of got that head and the stick here, what they're seeing is, so this red and also the yellows here are, those ATP synthase actually form pairs, like a V8 engine, and then they run in rows, like there's a lot of ordered structure in there. And so that's an interesting detail. That's what I'll be working on. Now, I'm just going to race through, and this is actually not the most exciting stuff of what we've encountered, what we're reconstructing. But I can, in fact, some of it I can't talk about because I can't let it out what we're doing. But this I can because it's a well-popular area to describe. I'll be publishing all of this work by the middle of this year in lots of different formats. All right, so this is our current work in progress for ATP synthase. I was hoping to actually show this to you live as it's running on my machine. I've got a gaming PC with all sorts of fancy lights, as every teenager would love, sitting on my desk, and it's running this world. But this is a movie that we've generated, so it's essentially a capture of that. And this is at 4K, so at the high resolution your TV could be these days. This is all running easily on a gaming machine. So we have... this is your ATP synthase. It's forming these pairs. This is a molecular world. The white dots are actually hydrogen atoms, but let's just focus on ATP synthase. I've isolated it just out. And what it is a rotary machine that's manufacturing ATP, which is that chemical fuel. So you got phosphate and ADP coming in, and they're being mashed together and forced together to form ATP. There's actually 3 spots where ATP are forming. We're now looking down on top of ATP synthase. And in turn, ATP is forming. It's a stepwise 120-degree rotation where ATP is being generated, step by step by step. Those shape changes in the head that are doing the manufacturing ATP are driven by this asymmetrical axle. There's a hydrogen pump that's driving this, and what's going on is there's loads more hydrogen atoms in here, protons down here, than outside, and so they're like a water wheel. They're trying to push their way through, which is driving that axle that drives ATP synthesis. Okay, so this is an accurate reconstruction, and now this is hinting to where we're going. Is I can populate this world with as many of these machines as I want now, and this is just. This is probably the least interesting engine of this whole respiration engine system I can show you right now. It's a tip of the iceberg, gives you a sense of where we're going. So, and I'll see if I've got any. So I was going to do a live demo. Unfortunately, my machine is busy doing a task I gave it too late today, and it's still chewing on that. So what it's doing is assembling the scene and getting it all organized, and so that in an hour or so I'll load it, and we'll have that world. The next iteration of it, populated live. I can press play. It'll play. We can have that camera move like you just saw there, wandering around. I can stop that. we can then take over, and like you would in virtual reality or in a video game, I can look around and go, hey, look at that, and we can go over and have a look and interrogate it. That's all going to be really easy and really normal. And ultimately, what I'm actually targeting is mobile phones, because every kid has a mobile phone in their pocket. They're immensely capable and incredible piece of technology nowadays. And so we're going to have these things playing live on a mobile phone through augmented reality, which is where you look through your phone and that ATP synthase will appear here on the desk, and students will be able to stand around the desk together and look at it and say, hey, look at it, and they'll see it live playing through their phones. It'll integrate the world we see around us with the ATP engine. So that's certainly coming really soon. I'll just play a couple of other factoids to do with ATP synthase. So this is an early model when I imported the X-ray crystallography, the actual model of ATP synthase. And what I've been showing you has been greatly slowed down so you can see all the different steps and chemistry that's going on. Now, this is actually in real time. It's producing about 100 ATP, these yellow boomerangs there are the ATP. This is a very primitive early visualization I was doing. But you see the rotary motor. What I like about it is it's not too obscenely fast that it's unimaginably fast for a human to watch. It is something we can watch in action, and you do get a sense of how fast it operates. But it's producing 100 ATP per second, and that's in a very optimal situation. So it's probably a lot less than that in most cells. And what's also interesting is this gear here that's running around the motor in different species and in different cells in different species, that gear can be made larger or smaller to regulate the speed and the amount of force. So at the moment, it's actually 3 of these protons coming through that drives the mechanical energy of ATP being generated. And one last one, again, so that was the real-time engine. And what you're going to see is I showed you the tubular membranes. These are the ATP synthase is here and here, those pairs, and we're looking like a slice through a cucumber down the center of the ATP of this mitochondrial membrane. And these are the engines, the molecular engines that are pumping, taking these little white dots, the protons, the hydrogen atoms, hydrogen ions, and pumping them in. So there's a much higher concentration here, and then they try and get out there, and that's what's driving. So this is the machinery for pumping hydrogen ions in, and then as they escape through this rotary engine, that's what's driving this mechanism. And this is in real time, or hopefully it will be. Nope. Hopefully it'll play. So this is the machinery all working in real time. So you have all the ATP synthase being generated in real time in these speeding rotary engines. And this machinery here is actually working to grab any hydrogen atoms there and pump them across the membrane. So, and it's actually, there's a whole bunch of other things that are going to be populating the world around there. The citric acid cycle, that if anyone who remembers from the high school or university level biology, the citric acid cycle is the most diabolically evil bit of chemistry that every kid has to learn. Trust me, I have studied for years. I never understood it. Now I do, and of course, it's made up of fantastic machinery that I can't wait to show you, but I can't just yet because I don't think anyone's actually put it together like we've done. So anyway, with that, I will stop and just give you a taste of what we've been doing. So thank you.
Thank you. We have time for a couple of questions. Yeah. And then either.
I was just so interested about. I was wondering because for all of the stuff you just showed to us, are they like separate videos, animations, or you do have sort of a database? I mean, like
So yes, so that's what we're doing is building like a video game, an asset database. So this is being developed for the Howard Hughes Medical Institute, who released these animations for free, and they're going to have dozens of. It's actually the problem now because all this stuff just runs. I can just start recording anytime I want. If you want a 30-second animation, you want a 3, it doesn't matter anymore. Like I can run it as long as you want. I'm more interested when we get to this where it's going to be live, real time. There's not going to be any movies anymore. It's just going to play, and you can take your time and explore it as long as you want. Because all this stuff plays endlessly. So we're going to create movies that, because that's going to be the normal, natural way for classroom use, is for videos for the teacher to use. Students will have it on their laptops. We're also developing touch tables for museums and science centers where it'll play on a big screen. You can interrogate in various ways. I'm really interested in getting it. We're going to have, by middle of this year, on augmented reality, these engines playing like I described it. That's going well, but it's all of it's game technology. But yes, I'm building an asset database, and each of my different clients want to have different types of content. So the American Natural History Museum are building a virtual room that you'll go into, and they want to be enveloped in this world, and I can do that. If the Dome people want to have Dome content 360, it's hard, but I can do that. So it's no longer about generating movies. It's generating a database of this stuff and building these worlds, just like the video game I showed you before. We can then have a different experience too. These are not linear movies of 10 or 20 seconds that you watch and you can watch it again, that's it. These are worlds that you can watch endlessly, or make looping GIFs out of, or become active real-time worlds. And that's going to work. It's happening now, and it's going to get really easy real soon.
And I was wondering, is that possible to do something like an app that you can just zoom in, zoom out? You start with some like human body, and you can just keep zooming to see any of sort of.
I've done a lot of powers 10 zooms. The problem is the scale of data is magnitudes each time. So powers of 10 zooms are, everyone desires to do one as an interactive piece. But there are problems with data, so I tend to do them as movies. I have done powers of 10 zooms. I've done at least 6 or 8 of them, where it goes from our experience of the world all the way down to the atomic scale and back out again. They're extremely difficult. They're the hardest thing I can do. We're developing this technology so it will do it in real time, but that's the next phase of this.
So context technology is still pretty hard to.
We just haven't developed it. It's certainly feasible.
Okay. Just a question on like the language that you used and the extended metaphor. Basically V8 engine, axle, rotary, manufacturing fuels, rotors, pumps, machines, gears, etc. Is that a conscious or a philosophical or a decision that is just emerged, or can you explain that using that metaphor?
First of all, those metaphors appear in the scientific literature. It's the way the scientists are describing these things, and they are metaphors or analogies that are valid in a lot of cases. I think, and also I am trying to avoid using any technical language in my presentation. I use a little bit then. I tend typically, when speaking to the public, I do not allow any scientific words to slip my mouth. You'll name something, but that's it. And you have to find language that's appropriate and meaningful for people. So yes, I get accused of that, but I think it's worthwhile to use, because I don't think it's that far off base. Maybe certainly I have a lot of problems with intelligent design people who think that, it's obviously been, I created this, okay? But it's a representation, giving you an insight. It's a sketch, a very, as much accurate as I can make it, but a sketch of what we know is going on. And I'm communicating to you as an artist in that sense, trying to give you a sense of that world, and I will use that kind of language in describing it. But whether that gives you a accurate point of view from the language I'm using, there's problems, I know, but I think it's still the best way, personally.
No, that's useful.
Jaden?
What's the most challenging aspect, I guess, of 3 aspects being first, understanding yourself, the mental model of what you're trying to graphically represent, especially if no one's ever represented it accurately before. Is that the most challenging aspect of production, or is it the actual sort of software engineering of the physics engine, or is it the engineering of the graphics itself? Because honestly, it doesn't look that complicated graphics wise. I mean, it's just colors and spheres and things. Is it the physics that's computationally intense?
No, it's more, so in this scene here you have 10 million points, atoms, and they're all interacting in various ways and with shadows cast, and it's just a lot
in that picture there's 10 million.
Yeah. And then this is not a completed world yet. This is not where, So, but the thing is, there are instancing, there's tricks we're using so that this object here is actually identical to that object there. So the memory management issue of having 10 million atoms, like the membranes themselves are this used to be the biggest technical nightmare to do is membranes, because they're fluid, like the surface bubble, you can imagine all moving around, and it's very dynamic. That used to be a hard thing to do. Now I do have a button, because my programmer has developed, click it and it's done. It's fantastic. So these issues are going away. But as far as productions go, at least a third or half of my year is simply spent where I'm surfing the journals, reading literature, going for long walks, trying to reflect on what I'm reading. But the thing is, everything I show is just scratching the surface of what's actually in the scientific literature, what's actually been discovered. But it's buried in technical jargon, and it's written in words, with the acronyms and all the fancy language they use in papers, and then they'll do a very crude diagram to represent their thinking of how it comes together. But then I can go access the accurate, the PDB, the X-ray crystallography of a molecule, and so the scientists may say this blob represents one protein, and here then you import it and you realize actually this protein is gigantic, this one's really small, so this diagram is completely wrong because they haven't really thought it through.
Do you sometimes feel an insecurity that you might be embarrassed by visualizing it incorrectly?
No, never, because I'm just reflecting what's actually in the literature, and it's really, this is pretty basic stuff in a lot of ways compared to what we know, and I do have to make, there's only fragments of our very exquisite detail we can understand, and there's large segments where we don't know, but we can speculate, have an informed speculation, and I will take the expert review paper, and they will typically bits they don't know, they'll say we don't know, and they'll do an informed speculation, and I can then represent that. So it's sort of the best of our, it's just a sketch, but it's the best we can do, right? It's the best representation we can do right now. But the thing is, it's barely scratching the surface of what we have. This is all 101 stuff, biology, and that's why I have to keep going on these themes. But actually the complexity of signaling and the way cells are talking to each other and how all your life comes about, let alone what's going on inside the cell, is just mind-blowing. So if you're interested in any of that, go to WEHI.tv to see all of our animations. A lot of it's DNA or molecular structures like this, or diseases like various forms of cancer or diabetes, and we have all our animations to give you a sense of what we're really learning about that.
Just, I think we're running out of time. Let this be the quick last question.
Would you, because of your work's very scientific in nature, because you're actually going through the literature, you're looking at the models, you're actually probably looking at it going, that's not right, and then creating models that you think are more accurate. Would you consider yourself more an artist or a researcher?
I'm an artist in a sense, but I'm trying to authentically represent what's actually. I don't make up models. I don't speculate. I will have a go. Well, what's going on there? And I'll go, and I almost always will find in the literature, in the discussion section, where they come to that bit where they say, we don't know at this point, but this is what we think. I'll go, well, if that's what you're thinking, I'm going to go with that. And it's just best we know right now, and things do change. But really, this is all still representing science from the 1990s, early 2000s. It's just exploding in our rate of discovery right now. The capability of technology in medical research is unbelievable, and we have just massive amounts of stories to be told.
So who owns those as well? Do you own your own work?
Most of my work is commissioned by philanthropies, and they own it, but they allow it to be used at no cost for various things.
So a school could buy your work, let's say?
Not buy, we give it to them for free, we distribute it. In fact, that's the key purpose of me interacting and collaborating with various organizations, is they invest millions of dollars in distribution. So the Howard Hughes will spend $5 million a year targeting schools around the US, and they're delighted if that same resource then gets used in Australia, and I bring that to here. And in Sweden, they're targeting 2.5 million school kids on a science excursion in domes. Around the world, there's 100 million school kids on science excursion, so they're happy for it to then go that way. So I work on lots of different projects. Most of it is free public education.
Okay, thanks.
Okay.